Eleanor Ewins from University of Groningen in the Netherlands. So, hi Eleanor, if you could
hi.
Turn on put your presentation on, please.
Yeah. Okay. So do you hear me? I switched over to headphones, so I just
yes, we do.
Sure. Okay, yeah. Great. Okay, so for me, it is good evening. I'm in the Netherlands, so it's 7:30 here. I want to talk today about a project I've been working on in our group, which, as you can see here, is a minimal pathway for the regeneration of redox cofactors. So quite different from the previous talk. This will be a synthetic biology, biochemistry talk. So the way we have approached this, hang on, I'll just turn on my pointer. The way we have approached this project is in the context of building a synthetic cell. And one might imagine that when you are constructing a synthetic cell, there are certain aspects of a real cell that you might want to consider and you might want to have included. So such as energy generation, compartmentalization, among others. And the aspect that we are focusing on and that we are exploring here today is the ability to control the redox state of the cell. And the goal of this project, let me just move this so I can see. The goal of our project was to construct a minimal synthetic system to provide reducing power to our synthetic cell. Can I go forward? Yes, thank you. And in order to do this, we based our system on nicotine amide cofactors, and the two main cofactors are NADH and NADPH, and you can see the names at the bottom of the screen. And these cofactors are really important for multiple different processes. So for NADH, this is involved in glucose breakdown, fatty acid oxidation. NADPH is involved in fatty acid synthesis, DNA synthesis, and these are just examples of the hundreds of cellular reactions that these two cofactors are involved in. So they are vitally important, and they have also been conserved across evolution. Conserved throughout evolution. So we decided this was a very good place to start constructing our redox system. So first of all, who I want to measure, rather, not what, is a PhD student in our group, Michaela, and he is the biochemist to my biophysics knowledge. So he is the one who did all of the protein synthesis, a lot of the small LUV work, and he also devised the metabolic system himself. And this is the system I'm going to be discussing today. I will go through each of the reactions in detail as we go along, but what I want to draw your attention to now is that it is composed of 3 cytosolic proteins, so soluble proteins that we encapsulate inside compartments, and also that the transfer of electrons between these cofactors cascading down is initiated by the membrane permeation of formate. So for the first reaction, this is the reduction of NAD to NADH, which is catalyzed by a formate dehydrogenase protein. And this happens when formate is added. And a nice way that we are able to visualize this reaction actually is due to the differences in signal between the fluorescent signal between NADH and the oxidized version NAD. So what we see is when NADH starts to form, we have an increase in fluorescence in this range. So the first way we approached this was to compartmentalize this first reaction inside large unilamellar vesicles. And then what we saw was a dependence on the concentration of the formic acid, which is the one here that starts the reaction. As one might expect, as the concentration of formic acid increases, the rate of the reaction also increases. And from this, we were able to determine the Km of this protein for formate. And we also performed similar assays to determine the Km of this protein for NAD. And then what we wanted to do is to scale up the system to GUVs, giant unilamellar vesicles. And one of the reasons to do this is predominantly because of their size. So due to their much larger size, we're able to visualize this reaction on a microscope, and it also provides more space for encapsulation of other components. And the methods that I used for this to observe the giant vesicles was using a microfluidic device, and this is just part of a drawing which demonstrates how it works. So inside channels, there are PDMS posts that form something resembling traps, and then we can trap individual populations of GUVs, which allows us to exchange the external solution. So then we can add our formate and watch the reaction live. So this is a confocal microscopy video you will see in a minute. There's a video. So this is our population of trapped GUVs in one of these small buckets. And then what you will see is as we flow in the formate, we see an increase in the luminal signal from the formation of NADH as NAD is reduced. So if we look at the fluorescent traces over time for these populations of GUVs, we see that comparable, not on time scale, but in behavior, we see an increase in the NADH being formed. And something also very unique, one might say, about this method is because we can continuously exchange the solution, we actually are able to modulate the rate of formation of this cofactor by consecutively changing whether the formate is present in the external solution or not, which would be very useful for modulating the concentration of a cofactor, for example, for subsequent reactions that use this. Then we went on to look at the next coupled reaction. So this depends upon the formation of NADH, and then a soluble transhydrogenase re-oxidizes the NADH back to NAD, while at the same time reducing NADP plus to NADPH. And in order to visualize the formation of NADPH, we actually utilized a protein sensor, which was specific, or mostly specific for this cofactor, as fluorescently these two molecules are indistinguishable. So this allows us to see that NADPH is being formed. So first of all, we put our system inside LUVs, and what you can see here is when the sensor is present and we have the reaction just with the FDH reaction, or we have the coupled SDHA-FDH reaction here, then we see an increase in the formation of NADPH. And this is a ratiometric sensor, so it works as one region of the spectrum will go down and one region will go up in terms of intensity. And here, again, we were also able to determine the Km for NADH and for NADP plus for the soluble transhydrogenase. And again, we moved the system into giant vesicles. And as I just said, as this is a ratiometric dye, we first start off with the signal between two channels, in this case, the 405 and the 488, due to the presence of the sensor. And then as we add in our formate again to start the reaction, we see that this ratio changes over time as more NADPH is being produced inside the giant vesicles. And then what we went on to do is to couple a further reaction to this synthetic pathway. And this was with the aim of producing reduced glutathione. And glutathione is an extremely important molecule in biology, in cells. It's responsible for neutralizing reactive oxygen species protecting and regulating thioproteins, detoxification, and has a role in the nitric oxide cycle, among many other things. So this is a very interesting and useful compound that we can produce, and that could then also be used in subsequent reactions. So I cannot see what is on the bottom of my graph, but first of all, what we did was encapsulated just this reaction, and then directly with NADPH, and then we see the formation of the reduced glutathione, and this was done in solution using Elman's reagent. And then what we see when we then couple this reaction to the rest of the redox pathway is that we have a dependence on the concentration of the soluble transhydrogenase that is included, on the rate of the reduced glutathione that is produced. And in these two bars, we see that there is some indirect or affinity for NADH, so the reaction will still run, but much slower when the when no NADP is present. But then when we remove both of the cofactors, we see that virtually, well, no reduced glutathione forms. And we could also put this reaction into LUVs, and what we see with the INAP sensor that I showed before is the formation of NADH. So in the absence of this part of the pathway, we have this formation of the NADPH, and then when we include the GORE and the glutathione disulfide, what we see is that the electrons are directly shuttled to reduce glutathione, so we do not have this formation of NADPH that's shown here. So this is why this this line is flat. And then what this graph shows is this is the red and the green are the same as before. So we for this concentration up until a certain time when the pool of GSSG is used up, then the reaction proceeds to start reducing NADP to NADPH again. But interestingly, what we see is there is a sort of a threshold concentration here, above which there is just such an excess of the glutathione disulfide that the electrons are just constantly shuttled to produce reduced glutathione. And this was performed in solution with the INAP sensor. So to summarize and to also look out to what we would like to do in the future, we were able to design and a minimal enzymatic pathway to modulate the redox state of these cofactors. And that this was also initiated by the addition of an external trigger substrate. And this is also useful in the context of synthetic biology, as we are not limited by the sometimes complex means of including a membrane protein, or the way that a membrane protein can slow down the translocation between of the substrate from the outside to the inside. We were also able to encapsulate this system in liposomes of different sizes, and we were therefore able to observe different types of behavior as a dependence on size. And we were able to produce reduced glutathione, which is a crucial biological compound. And in the future, what we would like to do is to further couple this system to other synthetic modules, so for example, to produce ATP, or to cause the cell to divide, to use compartmentalization. These are just some of the ideas that we are working towards at the moment. And with that, I would like to thank, of course, my supervisor, who is Dirk-Jan Slotboom, and Professor Pullman was also very closely involved in this project. Professor Marco Freyer also works at the same university as us and provided very useful insights into the action of the soluble transhydrogenase. And Professor Yi Yang very kindly provided the sensor, the INAP sensor, or the plasmid for the INAP sensor so that we could purify. And of course, I would like to thank the funding and the opportunity to present here, and all of you for your attention.
Thank you, Eleanor, for keeping on time as well, for your great presentation. I believe we have some questions.
We have one from Dr. Watts. Maybe Dr. Watts wants to speak, to open his microphone, or if not, I can read it.
Sure. Hi, Eleanor. Good to hear from you.
Yeah.
My question is, it's an interesting focus for a synthetic cell, but why is the time scale for the NADPH plus reduction different for LUVs compared to GUVs in the microcell? You didn't explain that. Why?
I didn't explain because I have time. We are not entirely sure. We don't want to pinpoint it. Of course, we discuss it. One of the reasons could be that we have quite different surface to volume ratios.
Yeah.
And also the difference in encapsulation between. So yeah, that would affect the permeation of the formate. Also, we would have between the LUVs and the GUVs, we'll have quite different membrane curvature. So this could perhaps also affect the permeation as well. They're prepared by different methods. I don't know how then this affects the permeation across GUVs that are, for example, prepared by a gel swelling method, and LUVs that are prepared by extrusion.
Is the lipid composition the same?
Yeah, the lipid composition is the same. The pH is slightly different. 7.5, but it really shouldn't make a difference.
No,
especially the first protein is basically has the same behavior between pH 4 and pH 10, sure. So it should be fine. I'm trying to think if there is something else.
Okay, it's an interesting focus on synthetic cell. That's good. Okay, give my best regards to Bert and to Derek.
I will.
Okay, thank you.
Are there any other questions?